Hey everybody, welcome to week 9. This week we are going to be going through more fastidious organisms. And in this case, all of the fastidious organisms are going to be gram-negative rods. Last week we discussed the gram-negative coxine fastidious organisms of Neisseria and Moraxella catarrhalis. This week is all about the fastidious gram-neg rods. So I'm going to just pop up the PowerPoint here for you guys. Let me get that into slideshow. All right, so the very first group of gram-neg rods that we're going to talk about out of this chapter is going to be Haemophilus. These are a huge group of pathogens, and I've got 4 listed there that I want you to be aware of. So we have Haemophilus influenzae, Haemophilus parainfluenzae, Haemophilus ducreyi, and Haemophilus aprophilus. All right, so in general, again, they are all gram-neg rods. They are actually oxidase variable. Remember last week I mentioned that whenever we get a gram-negative, we always oxidase test right away. So when you go to oxidase test, you will see that Haemophilus influenza and Haemophilus parainfluenzae will be oxidase positive. Very much, definitely should remember that. Whereas some of the others are going to be more oxidase negative. Again, these are gram-neg rods, and in fact, they're very tiny, tiny gram-neg rods. They are not going to jump out at you by any means. They are small. A lot of times people call them coccobacilli. They don't tend to look truly rod-like all the time. They kind of look like that oval shape instead. I often call these junk in the background. They are easy to miss because they are so small and they just kind of are fainter. They just don't stand out at all. So you have to have a good eye for these. Now, the other thing with these is I said they're fastidious, and these are fastidious because they require extra nutrients, extra stuff to grow. And in the case of Haemophilus, especially Haemophilus influenzae, they require what we call X and V factors for growth. X stands for hemin, which is blood, and the word Haemophilus itself actually means blood lover. So they love its blood, and then the NAD is the V factor. So pretty much the Haemophilus will need either one or the other or both, except for Haemophilus aprophilus, or you can call it paraphilus. That doesn't need either one. So I'll show you that. I think I have that listed in a chart here. And then also these Haemophilus tend to inhabit the upper respiratory tract with the exception of Haemophilus ducreyi. Haemophilus ducreyi is actually an STD, which we're going to see. Okay, so there's the gram stain. Now this is a nice brighter looking gram-neg rod, Haemophilus gram stain. They're not always this wonderfully stained, but you can look at it and see, look at the shape. And you can tell they're not a really nice rod shape. They're kind of more of that oval coccobacilli shape. So you can kind of get that feel here from this picture. All right, so we are going to start with Haemophilus influenzae. It is the biggest pathogen out of the bunch. It is the one that goes along with some significant diseases. There are 6 different serotypes of Haemophilus influenzae, and they tend to label them A through F. And then there's other non-typable strains that just aren't typable into a serotype necessarily. Now, Haemophilus influenzae doesn't necessarily cause influenza. That's where it gets really tricky because influenza is a respiratory illness. But remember, influenza is caused by influenza viruses. We're talking about bacteria. So there is lots of other respiratory diseases Haemophilus influenzae causes, just not necessarily the influenza everybody thinks about. Again, very much spread via respiratory droplet, person to person. So here are some of the diseases listed there that it is known to cause. Meningitis is right there at the top, and that has been specifically linked with the serotype B. So it's called Haemophilus influenzae type B, or HIB for short. This used to be the #1 cause of meningitis in young children underneath the age of 5. They have since come out with a vaccine, and that vaccine has really whittled down how many children are suffering from this. So very few are getting meningitis versus how common it used to be. Now, of course, there's always people that don't vaccinate their children, and they're still going to be at risk. Haemophilus influenzae also causes pneumonia, septicemia, bacteremias. It is the #1 cause of acute epiglottitis. If you don't remember, epiglottis is that little flap that closes over your airway. And so when you get epiglottitis, what that means is that has become inflamed. And when you inflame that little flap over the airway, obviously it's going to lead to breathing difficulty. It's going to obstruct your airway, which can be very dangerous. So Haemophilus influenzae is the #1 cause of acute epiglottitis. It is also a cause of pink eye. You can also call that conjunctivitis, and that is specifically Haemophilus aegypticus. So it used to be Haemophilus influenzae subspecies aegypticus. I think they might have changed the name to just Haemophilus aegypticus now, but if you ever reference readings or tests or anything that says Haemophilus influenzae subspecies aegypticus, that's what's referring to the pink eye part specifically. All right, so that is showing the different percentages of diseases caused by Haemophilus influenzae. As you can see there, 58% now has been linked with pneumonia. So this is basically going off of when people do have Haemophilus influenzae, which type of illness did they get with it. So the majority of the time people are having Haemophilus influenzae infection, it's due to pneumonia. Before the Hib vaccination, this little 7% meningitis would have been a huge portion of this pie chart, but because that Hib vaccination has been out and working wonders, you can see it whittled it down to 7%, which is pretty freaking amazing. And then the other 4% would be like your acute epiglottitis, your conjunctivitis, that kind of stuff. All right, Haemophilus parainfluenzae is also a cause of upper respiratory infections such as otitis media, which is ear infections, and conjunctivitis as well. Can also sometimes be seen with pneumonia. Now, there is a group of bacteria called the Haysick group, and these are 5 different organisms, each with those initials, that have been grouped together as they all cause slowly progressive endocarditis, so all about the heart. So I'll say that again. Haysick is a group of bacteria that have been put together because they all cause slowly progressive endocarditis. Now, the H in Haysick stands for Haemophilus. Haemophilus aprophilus used to be the main organism I would always teach that was part of the Haysick group, and now they're saying that there's other Haemophilus that can also be part of this. So the H tends to just stand for Haemophilus species in general. So Haemophilus parainfluenzae could be that H in Haysick at some times. But again, in my testing, my quizzes and exams, I'll probably reference it more as Haemophilus aprophilus. But if you were to ever have to write out what each of those initials stands for, and we're going to learn all of them, you just should write out the H as Haemophilus. Okay, so Haemophilus ducreyi is one of the oxidase negative organisms. So again, Haemophilus influenzae and Haemophilus parainfluenzae were both oxidase positive. Haemophilus ducreyi is oxidase negative, and again, this is our STD. And it's known to cause what we call a chancroid, which you can see in the picture, which is just a nasty ulcer of the genitalia. And then it says defined as ulceration of lymph nodes in the groin as well. So obviously passed sexually. Okay, and media. Media, again, fastidious, it's going to grow on chocolate. It will not grow on blood agar because chocolate is going to give it both the X and the V factor for Haemophilus to want to grow. So chocolate is a hemolyzed blood agar, so that's why it's called chocolate agar. It's actually blood agar that's been hemolyzed. So the hemolysis of those cells let out the heme in the X factor, and then it also has additional NAD there. Blood agar does not contain NAD, it only contains the blood, so that's why they don't like to grow on blood agar, except when Staph aureus is present on blood agar. There is a thing called satelliting that Haemophilus is known to do. Whenever Staph aureus is present on blood agar and Haemophilus is part of the infection or part present as well, you will see Haemophilus growing around the Staph aureus colonies, kind of like little colonies around the main Staph aureus colonies, and that's what we call satelliting. And the reason it can do that is that the blood agar does have the hemin, and when the Staph aureus grows, it hemolyzes those blood cells to also release out the NAD so that both hemin and NAD are present at that point, and then Haemophilus will grow as well. So that's something you need to kind of remember. And then, of course, MacConkey, yes, these are gram-negative rods, but again, they're very fastidious, so they will not grow on MacConkey. And here it's kind of showing the satellitism. in general, by the way, Haemophilus tend to be smaller gray colonies, as you can see here, nothing fabulous. If you look here, these are Staph aureus. They're beta hemolytic, creamier, larger colonies. And then the Haemophilus, which are kind of growing around it, are the smaller gray ones. So again, they can do that because the Staph aureus hemolysis has released extra NAD out, so now it wants to thrive and grow. So here's the ones that need X's and V factors. You should definitely remember this. These are actually tests that we can run in the lab. Different labs can use what we call quad plates that have X and V factor in it, and you see which they grow in. So as you can see here, Haemophilus influenzae have to have both X and V factor to grow. So if I were going to perform a test in the lab and streak out my unknown organism, say I think it's a Haemophilus, and I'll put it onto a plate. On one side it has the X factor, on the other it has the V. If it grows on both, if it grows only where there's both X and V, then that tells me it's more influenzae. If it can't grow on just X only or on V only, then it can't be. We can also use little discs. You know how you guys have been playing with the Optikin and bacitracin little discs? There are actually X and V discs as well that you can grow or use and plate onto agar. So there's different ways that you can test to see how does this grow. Does it respond by growing with the presence of both? So in that case, that would be influenza. It has to have both present to grow. If it's para-influenzae, it only needs the V factor to grow. It doesn't need the X factor. Ducreyi only needs the X factor to grow. It does not need the V factor. And then as I said before, Aprophilus or Paraphrophilus doesn't need either one, neither X or V. So definitely remember which ones need what, and these are part of the testing workup that we can do in the lab to help identify what Haemophilus we would have present. So in general, it would go gram stain. We'd see it's a small gram-neg rod or gram-neg coccobacilli. We would oxidase test it. That would kind of start to give a clue. Again, Haemophilus influenzae and para-influenzae are oxidase positive, and the other two tend to be oxidase negative. And then we could use X and V factors as well to help test to determine what we have. So that's all part of the workup of Haemophilus. Now, I will give you a little trick. I always remember that influenzae always needs both X and V. It's my main pathogen out of the four. The way that I remember the Ducreyi and the para-influenzae part is that I know Haemophilus ducreyi is an STD. And so whenever you think XXX, the triple X, you always think porn shops, and that kind of goes with STDs. So that's how I always remember Ducreyi needs just the X factor. And then I know para-influenzae is the opposite. So that's kind of how I keep them all straight in my head. Now, you probably are wondering why we haven't, I'm just kind of seeing what's here, why we haven't really discussed Haemophilus paraphrophilus. We really did back on this slide. That's the Aprophilus. Again, it's just known to be linked with slowly progressive endocarditis. That's all you need to remember on that. And it does not need either X or V factor to grow. So that's kind of the brief mention on that one. We didn't have a slide set apart for that specifically. Okay, this is just a little flow chart on Haemophilus. Again, because Haemophilus is associated with meningitis, you could be getting a CSF sample. It's also associated with septicemia, blood culture. If it's a pneumonia, you're going to get a sputum culture. You know, it just depends on what disease they have or whatever. And so it's just showing you how they work up. They inoculate it. They see the gray colonies. Again, they would gram stain it. They would see it's a gram negative. They would oxidase test if it's oxidase positive. Then they could do the X and the V factor. If it requires both X and V, then it's Haemophilus influenzae, and they can serotype it to see what specific serotype is present. Because remember, there's A through F serotypes. If it's oxidase negative, it cannot be a Haemophilus influenzae then. So it's just a quick mention. Here's the gram stain part over here. So it's just a quick showing of how they use that exact testing that we just discussed. All right, moving on to other fastidious gram-neg rods. Here are the rest. We will be going through that Haystack grouping right now. So there's where it's listed, a group of organisms that cause slowly progressive endocarditis. They are all gram-neg rods. They all love CO2, capnophilic, to grow. Again, they're fastidious. They will not grow in blood agar. They're typically slow-growing organisms. They're not like the quickest. And then all tend to be normal flora of the oral cavity. So they're very similar in characteristics. That's why we have all grouped them together under the name HASIC. So here are our members. Again, H has been thought to now just stand for Haemophilus species in general, or you can know it as Haemophilus aprophilus or Paraphilus. Just so you guys know, in the microbiology world, they love to rename organisms, and it drives me crazy. Like forever, I have always known Haemophilus aprophilus to be Haemophilus aprophilus. Now they've decided to rename it, and it's like, drives me nuts. They do it all the time. All of a sudden, you'll have learned one thing in college, and like 5 years down the road, it's a totally different name. It's like, why? So that's why if you keep hearing me mention it 2 ways, that's why. All right, the A is a lovely name. It's Aggregata. Oh my God. And this changed names too. This used to be something different, but it's Aggregatibacter actinomycetum and Aggregatibacter aprophilus. C is Cardiobacterium hominis, E is Eikenella corridans, and then the K is Kingella. Thank God for the simplicity of that one. I will warn you, one of my favorite questions to ask on tests in general is a short answer. List me the HASIC group names and what they all are grouped together for. So you would have to list these 5 out. Now, do I expect you guys to be spelling experts? No, but I expect you to list them out in a way that I can read them to know that, okay, yeah, you got what you're talking about. You would be able to pick this out if you could. So spell them in such a way that I'm going to get the gist of what you're trying to spell out. So just to keep that in mind. A little bit more on a few of these HASIC groups. Eikenella corridans is a gram-negative rod, of course. It has been known when it grows on the agar plate to pict the agar, meaning it kind of digs down into it, makes it like a little depression. It has a bleach smell. I always like to think of corridans meaning corrosive, and bleach is corrosive. That's how I kind of remember that. Eikenella corridans is oxidase positive, and it has, besides being linked with the slowly progressive endocarditis here, it has also been linked with bite wounds or fist wounds. So again, because it's carried in the oral cavity and somebody punches someone else in the mouth and say they get abrasion, and that oral, like normal flora of that person gets into that abrasion on their fist, they can get an Eikenella corridans infection. So usually seen with human bites or clenched fist wounds from punching somebody. Capnocytophagia is another gram-negative rod. That's a normal flora of the oral cavity. It's capnophilic. It tends to have a yellow-orange color to it on the agar plate, typically oxidase negative. This, because it's also found in the oral cavity, has been linked highly with dental plaque and periodontitis. Pastorella, again, we're on the gram-negative rod theme. This is oxidase positive. This is said to have a musty or mushroom smell. Pastorella multocida is the most common out of the group, and that's the one I want you to remember very much as Pastorella multocida. Very much a zoonotic disease, meaning this one is typically seen infections of like cat scratches, cat bites, dog bites. It's really linked with cats, but it can be linked with dogs and some other animals too. Anytime that I'm reading like a case study or test question, like on just say a review site, and they start asking about a gram-negative rod, oxidase positive, seen from a cat bite, I right away start thinking, oh, Pastorella multocida. I mean, there's other things, but I heavily start thinking Pastorella multocida on that one. Brucella. Brucellas are small gram-negative rods. They tend to be more coccobacilli, and they have all been a zoonotic disease. So you can see they're all linked with different animal hosts, so they are a zoonotic kind of a disease. They are also very much a bioterrorism type of bacteria. This is one that's on our bioterrorism list that we have to, anytime we grow Brucella, we have to report it out, send it off, that kind of thing. So you should remember the 4. Brucella abortus has been linked to cattle. Brucella melitensis is sheep and goats. Brucella sui is swine. That kind of goes together. And canis, of course, is dogs. In the U.S., you really don't see this unless people are still drinking like unpasteurized milk. But it's very much needing a biosafety level 3 to work it up. If you ever think this might be present, you should have a level 3. If you don't, you need to send it out immediately. And again, it is kind of a bioterrorism weapon. So it says that at the bottom of the screen here, used as a biologic weapon. So how do you get Brucella? So we've talked about the zoonotic, the animal host. So you can get it, of course, through different abrasions or breaks in your skin, like I said, unpasteurized milk or other infected food products, or it can be aerosolized through like bioterrorism weapons. You can see it if you're not working in a level 3 lab, you can get it that way. So what happens when people get a Brucella infection and why they love to use it for bioterrorism is it causes a pretty significant infection. It easily disseminates throughout the body. So it hits a lot of the major organ systems. It hits the lungs, the liver, spleen, CNS. I mean, it does its damage. So any part of those that it hits, it causes infection and inflammation of each part. So when it hits the heart, it can cause endocarditis. If it hits the joints, arthritis. It can do hepatitis in the liver, you know, that kind of thing. So you can see how each organ gets its own infection with it, encephalitis with the CNS system. So besides, obviously, fever, which goes along with a lot of different infections, it has actually like a relapsing fever, night sweats, muscle pain, myalgia is what muscle pain is, that kind of thing. So we would, of course, for specimen collection, blood is a good one to take here to see it in blood cultures. If we think it's infecting the bone, bone marrow, fluid from the joints, wherever or biopsies, whatever we think is being infected, the doctor can usually tell if something's inflamed by the symptoms, by the feeling on their abdomen, things like that. So they kind of have an idea where they would need to take from. And again, it likes extra CO2 here, and it's not the quickest grower. It's going to take a little while to grow. When it does grow, it's not really pigmented. It's non-hemolytic, very much oxidase positive gram-neg coccobacilli though. It is non-modal. There is a few other tests that can be run. There's lots of tests you can run on any bacteria. I try to simplify the amount of tests that you guys have to remember. In this case, it has the enzyme urease, that's urease positive, and it can reduce nitrite to nitrate. So it would be nitrate reduction positive. All right, another zoonotic disease that's also a biological weapon would be Francisella tularensis. I always think rabbits. The minute I hear this name, if any of you have ever heard of tularemia or Francisella tularensis, maybe you think of rodents and rabbits, but it causes the infection tularemia, which is very much carried by different rabbits, rodents, beavers, muskrats. There's the zoonotic part. So of course you get it by handling these different animals. So maybe you're somebody that's like a trapper and handles the skin of those animals, or if a deer fly bit the rabbit and then comes bites you, it could transmit the Francisella to you, things like that. So this causes nasty little lesions. It can cause them on the skin, and that would be called an ulceroglandular tularemia, which you see here on the thumb, that skin ulcer. But it can do the same kind of thing on the organs inside the body. So it can cause lesions and damage to your organs, like your liver and heart and stuff. So again, that's why they love to use it as a biological weapon, because it's hitting lots of different parts of you to cause massive damage. The other big thing they like with this one is it's extremely infectious. It doesn't need very much to be present to cause an infection. And so they quote usually just 10 organisms could be present. That's it. That's enough to be infectious. And that's very low. That's a low dose. That's not much at all. All right, symptoms with this: fever and chills, headache, cough, chest pain. Wherever it entered, it could have lesions at that site of entry. Kind of causes these ulcerative buboes similar to the plague. Sometimes it's a respiratory disease. This would be more seen, especially if it was a bioterrorism attack, because anytime we're thinking bioterrorism, they're usually making things inhalable. Like they want people to inhale it because that's the easiest way to transmit it to people. So if it shows up more respiratory, then we're kind of linking it more with the bioterrorism. So again, blood cultures, especially if we think somebody's become septic, and then any organ that's infected, or if it's a skin ulcer, we would culture from that lesion. It is also a biosafety level 3, so if you think you have this, you need to have a biosafety level 3 to work with it. Again, this is fastidious, so it's not going to grow in blood. It will grow in chocolate or another agar called BCYE. Takes a little longer to grow. It's another slow grower, in this case up to 48 hours. Again, another gram-negative coccobacilli, and it is oxidase negative and urease negative. Legionella, another gram-neg rod. In this case, these gram-neg rods are thinner and faint staining. They do not take stain very well at all. That is a feature I would remember about Legionella. They also are fastidious, so they're going to need that BCYE agar. That agar is actually specifically made for Legionella. I know we mentioned it here with Francisella, but I really want you to link BCYE agar with Legionella. It actually stands for buffered charcoal yeast extract. And in that agar, it's going to have iron and L-cysteine so that Legionella can grow. Legionella needs these two items in order for it to grow. So those two items are found within that BCYE, so please link that with Legionella. Out of all the Legionellas, Legionella pneumophila is our main pathogen. It has 15 different serotypes, but the main disease that Legionella pneumophila causes is a pneumonic-like illness. That's why it's a pneumophila, and it's something that we call Legionnaire's disease. And so you probably, some of you might have heard of this, but Legionnaire's disease was named because the first outbreak of this that they discovered and linked and it caused an outbreak was in the American Legion. And it was somewhere in the East Coast. It was like Philadelphia or somewhere like that. But it was at an American Legion and everybody fell ill with pneumonia, basically, kind of like a pneumonia. And it was because of an infected air conditioning system in that American Legion. And so when you look here at the reservoir, very much water is one, and it is hugely associated with air conditioning systems. Now you're all probably like, oh, I'm not turning my air conditioner on. Typically we're okay. My God, my air conditioner runs nonstop. I live in Phoenix, and I'm okay. But there is a chance. There was an outbreak of this last year, either last year or 2 years ago again. So when this does pop up, it typically is associated with outbreaks. So I think I will link an article in the announcements for that. I think if I still have it, I'll link that. But that's always interesting because it's still relevant. It's still seen. So you will isolate this from the respiratory tract, typically, because it is causing a pneumonia-type illness. You can directly detect it if you want through a fluorescent test, which is a popular thing to do. Here's the BCYE agar. It is buffered charcoal yeast extract, so it's charcoal. That's why it's black-looking agar. And then I think I will put a picture in the announcements at some point this week of the fluorescent antibody test that you do to visually see Legionella right away by looking for an antibody to it and using a fluorescent technique. So I will put that in the announcements. All right, Bordetella pertussis, oxidase positive, gram-negative rod, non-modal. This is the cause of whooping cough. I think we have all been vaccinated for this. This is part of that DTaP vaccine. Remember, we learned D was diphtheria. The P is pertussis for whooping cough. It has very selective media that we can use to isolate it. Bourdet Ginkgo is a good one. The Bourdet even looks like Bordetella. You can use a transfer system called Jones-Kendrick, but you can also use another agar called Reagan-Lowe. Typically, when we are collecting this from anybody that we suspect having whooping cough, we are doing a nasopharyngeal swab. So that is a very small typical swab that goes up the nose, quite high in it, and twirls up there. That's the same way that they collect influenza testing is a nasopharyngeal collection. So for Bordetella pertussis, same way, nasopharyngeal swab. Unless they're a really small child, because my daughter was tested for a different disease, but they had to take it a different way. So if they're a really small child, we would use something different to collect. Okay, that's a lot of info. I know there's a ton more organisms and test results to know this week than there was last week, but just sort through it as best you can. Keep track of which ones are oxidase positive, which ones are oxidase negative, because I know there was several in each area. Try to know the main disease that goes with, any other special notes on it, and just really dig into material, take the material out of the PowerPoint to learn it, put in flashcards, do something like that. Otherwise, you guys have a fabulous week, and reach out as always if you need anything. Thanks.